Good morning, everybody. My name is Cornelia, and I'm the product manager for gene editing tools here at Takara Bio Europe. So thanks for attending today's webinar. I'm very pleased to present some of our tools for gene editing today. So just some practical things before we start. So you will be muted during the webinar. However, you can ask questions at any time by typing into the question box, and we will have time at the end to discuss. This webinar will also be recorded, and we will provide you with a PDF copy of the slides at the end. So yeah, let's get started, and I hope you will enjoy. So today's talk will be divided into two parts. In the first part, I would like to give you a brief introduction about CRISPR-Cas gene editing. So we will see a bit the history of CRISPR-Cas, what it is, and how it can be exploited for genome editing. And then the second part will be a bit more practical, and I would like together with you discuss typical CRISPR-Cas workflow that I split into 5 steps. And we will discuss the following points, so choosing the target, designing guide RNAs, testing guide RNA efficacy in vitro, the delivery of Cas9 and guide RNAs to cells, and here especially I would like to present you our new Gesicle production system, and finally we will also see several options on how to monitor indels. Okay, just one comment. Apparently the sound is a bit low. Let me. Okay, I hope it's a bit better now. I'll try to speak up a bit. Okay, so as you may already know, CRISPR stands for clustered, regularly interspaced, short palindromic repeats. These so-called CRISPR arrays have been originally identified in bacteria, where it has been shown that bacteria have these, here shown as black diamonds, these repeated base areas in their genome. And it has also been shown that bacteria tend to integrate short pieces of foreign DNA into these CRISPR arrays. So for example, DNA from invading phages or plasmids. These CRISPR arrays are often located next to a so-called Cas locus, here shown on the left. And upon expression, the Cas locus will produce the so-called Cas endonuclease proteins. Upon expression of the CRISPR array, short so-called pre-CRISPR RNAs will be produced, and those will be further processed into the so-called CRISPR RNAs. And as you can see here, each CRISPR RNA has one fixed sequence part which comes from the repeat arrays, and then one variable part which is from the foreign DNA piece. And it has then been shown that the system is a very elegant system of adaptive immunity in bacteria, because if a phage, for example, infects the bacterium a second time, then this little CRISPR RNA will guide the Cas endonuclease protein to this foreign DNA thanks to this sequence homology. And the Cas protein will then cleave and destroy the foreign DNA. So the first reports of these CRISPR arrays have already been published back in 1987, but then it took 20 years to discover that this whole system is indeed a system of adaptive immunity. And I would say the whole breakthrough of this CRISPR-Cas technology is quite recent, and it dates back to 2011 and 2013, where it has been then shown that CRISPR systems can also be expressed in other bacteria, for example in E. coli. And finally, that also these CRISPR-Cas systems can be expressed in eukaryotic cells, and that they can be used for gene editing in those cells. And this has really been a real revolution in the field of gene editing. In 2014, there has also been a genome-wide functional screening using Cas9, and the crystal structure of Cas9 has been published, which gives, of course, further insights into the mechanism of the CRISPR-Cas gene editing. The applications of genome engineering are, of course, very broad. You can use CRISPR-Cas gene editing to create animal models, for example, knock-ins or knockouts. You can also just modify cells and culture. This is often the first step. And there's also a big application field in the area, in the biotech area, for example, in the field of development of new crops. And of course, there's big hope in the field of medicine to cure human disease using gene editing and gene therapy. So as I told you, Cas9 is an endonuclease which creates double-strand breaks in the DNA. And I would just like to go one step back to the basics, because these double-strand breaks are really very dangerous lesions for eukaryotic cells. And that's why cells have developed very efficient repair pathways. And as you can see here on this picture on the left, in response to double-strand breaks, for example, induced by irradiation, the cell will massively activate the repair machinery. This is shown here by these green dots. And eukaryotic cells have mainly two repair pathways to get rid of double-strand breaks. The first one is the non-homologous end joining pathway, and I would say in 95% of the cases the cell will go for this pathway because it's very rapid and flexible, and the cell really just needs to get rid of the double-strand break very quickly. But non-homologous end joining is error-prone, meaning that it will insert little insertions, deletions, or point mutations in this target site. And we will see later how this can be exploited for gene editing. The second repair pathway is the homologous recombination, and this is really the perfect repair. But as homologous recombination needs a repair template, it is restricted to some phases of the cell cycle. And that's why also in most of the cases the cell will go for the NHEJ pathway. So this is shown here again schematically. In response to a double-strand break, the cell has two possibilities: either the NHEJ pathway, which is error-prone and will create so-called indels in the target gene, or the cell can go for the homologous recombination if a repair template is present in the cell. So how can this be exploited for genome engineering? This is very easy. You just need to artificially create a double-strand break, and then the cell will do the rest and repair this double-strand break, either through the non-homologous end joining pathway, which can be used to create knockouts, or via homologous recombination, and then you can use this technology to insert any cassette you wish to do knock-ins. So of course you would not use irradiation to create the double-strand breaks, because irradiation would insert double-strand breaks everywhere in the genome. But in the past, people have already used enzymes to create site-specific double-strand breaks. For example, the so-called zinc finger nucleases and also talons. So these are enzymes which are composed of one DNA recognition domain and then the cutting domain here, which will induce the double-strand break. And then again, the cell will do the rest and repair this double-strand break using non-homologous end joining or homologous recombination. And you can use these enzymes to create knockouts or knock-ins. However, when using zinc finger nucleases and talons, there is one major restriction, because these enzymes use a protein domain to recognize the target site in the genomic DNA. So, and this means that each time you want to target a different genomic sequence, you would have to re-engineer the whole protein. And this is quite tricky, and not all labs are experts. So it would be really great if there was a nuclease which is targeted to a specific genomic sequence by RNA and not a protein domain. And then you could really change the target specificity very easy and at low cost by simply changing the RNA part. And this is the beauty of the CRISPR-Cas9 system, in which the Cas9 endonuclease protein is guided by a small so-called guide RNA to the genomic target sequence. So how does this guide RNA look like in a bit more detail? All guide RNAs have a fixed sequence here in blue, also called the scaffold sequence, and then 20 nucleotides of your target-specific sequence. Your target gene also needs to have the so-called PAM sequence, which is NGG for the Streptococcus pyogenes Cas9. So this is very frequent in the genome. And this PAM sequence allows then Cas9 and the guide RNA to recognize the target gene. And then thanks to the sequence homology of the guide RNA with the target sequence, this complex will be guided to your target gene, and the double-strand break will occur a few nucleotides upstream of the PAM sequence. And as these double-strand breaks are very dangerous for eukaryotic cells, the cell will quickly try to repair the double-strand break, and in most of the cases the non-homologous end-joining pathway will be used. And this is an error-prone pathway which creates indels in the target gene. And if, for example, this creates a frame shift, you can use this to create a knockout in your gene of interest. So let's move on to the second part. So again, on the right, I put the 5 steps of a typical CRISPR workflow I would like to discuss. And on the left, I put Takara CloneTech tools for gene editing. And each time when there is a corresponding kit, I would like to spend some time to explain how it works and where you can use it in the workflow. So once you have chosen your target gene, you will need to design your guide RNAs. And this is a very important step. So of course you could do this manually by identifying a PAM sequence in your target gene and then counting the 20 nucleotides upstream to define the target-specific guide RNA sequence here in red. But I would really rather recommend to use one of the existing online tools. There are many, many online tools that exist nowadays. I also put here a link to a list of available online tools in the presentation. And these tools will not only identify PAM sequences in your target gene and give you a list with possible guide RNAs, these online tools will also score the guide RNAs for the probability of off-target effects. And then you can really choose the guide RNAs which have the lowest probability to have also off-target effects. It might also be good to use different tools, because all these tools use different algorithms, so that you can check whether your favorite guide RNAs that are the best ones in one tool are also have good scores in another tool. Our R&D also realized that our best guide RNAs always contained a G in the first position and an A or a T in the 17th position. So the G at the first position, I believe it's important for good expression using U6 promoter. And for the A or the T, we don't have a real explanation, but it's just a fact that it was always present in our best guide RNAs. So now, once you have designed your guide RNAs, and I would recommend to design 3 or 4 guide RNAs at least per target gene, I would then recommend to test the guide RNA efficacy in vitro prior to the delivery into cells. And for this, we have the so-called guide RNA screening system that I would like to present you here. So this guide RNA screening system is composed of 2 kits. The first one is an in vitro transcription kit to produce your own guide RNAs. using T7 RNA polymerase. And the second kit is the so-called screening kit, which contains PCR reagents to amplify a part of your target gene. And this kit also contains recombinant Cas9 that you will use a bit like a restriction enzyme in this in vitro assay. And you will then simply incubate in a tube your guide RNA together with your PCR fragment of your target gene and the recombinant Cas9. So this is shown here. In the very first step, you will produce your guide RNAs, and then you will amplify part of your target gene containing the PAM sequence. So you will amplify about a 2 kb fragment. And then you will simply incubate in a tube the recombinant Cas9, which is provided in the kit, the guide RNA you have in vitro transcribed, and your PCR fragment. And if the guide RNA efficiently guides the Cas9 to your target gene, then you should see cleavage on the gel, which is shown here. So now you might ask yourself, why should I test my guide RNA in vitro? So I would say it's really a very quick and good pre-screening of your guide RNAs to pre-evaluate the efficiency for your target gene. And the most important point is that you will not waste time to clone and to transfect guide RNAs that don't work, because the guide RNAs that don't work in vitro, they will not work in your cells. So you can really choose and work with the best guide RNA. And another interesting point is also that you can compare efficiency of cleavage for potential off-target sites using this kit. So for example, if the in silico tools give you a guide RNA and you don't have the choice, and all the guide RNAs you can use have also a probable off-target site, then you could also PCR amplify this potential off-target and check in vitro whether there is a probability that the guide RNA will also guide Cas9 to this site. And this is, of course, what you would like to avoid. The screening kit is really, it's a very fast screening, and there are no cloning steps required. So in total, it's really a streamlined protocol which can help you saving time. The second question you might ask yourself is whether the guide RNAs that work best in vitro will also work best in cells, because it's a quite artificial in vitro assay. And here I would like to show you some data from our R&D which show a good correlation between both tests, in vitro and in cells. And these are data for the CXCR4 knockout. So our R&D designed 4 different guide RNAs and assessed them in the in vitro screening assay. And what you can see is that 3 out of 4 guide RNAs performed quite well in vitro. There are lots of cleavage fragments. But the guide RNA number 3 was not very efficient in vitro. You can see that there's quite a lot of non-cut fragment left. So our R&D then transfected those 4 guide RNAs and also Cas9 into HeLa cells, and they used a FITC conjugated antibody against CXCR4 to monitor the surface expression of the protein. So you can see here, in case of CXCR4 expression, you will get a fluorescent signal in the flow cytometer. And what you can also see here is this leftwards shift in the cell population when transfecting the guide RNA 1, 2, or 4. And this is an indication of a functional knockout of the CXCR4 protein. However, what you can also see is that when using the guide RNA 3, which was not efficient in vitro, our R&D observed that there was no shift in the cell population, and that this guide RNA was also not efficient in cells. And that's why I said that this in vitro screening kit is really a good tool to get rid of the inefficient guide RNAs and to work with the best ones. Okay, so once you have then chosen the best guide RNAs to deliver to your cells, there are really many options on how you can deliver Cas9 and your guide RNA into cells. So today I would like to focus on our new Gesicle production system, and I will not have time to discuss other options, but just to let you know, you can, if you have easy to transfect cells, you can, for example, use plasmid delivery. We also have systems for this. For in vivo delivery or hard to transfect cells, you could also choose AAV, so viral based delivery of Cas9 and the guide RNA. Or you can also choose the option to directly transfect Cas9 mRNA together with a guide RNA. But as I said, today I would like to present you our new Gesicle production system, which can be used to deliver active Cas9 protein together with your guide RNA to cells. So first of all, you might wonder what the name Gesicle stands for. So Gesicles, the name comes from vesicles and glycoprotein, because Gesicles are cell-derived nanovesicles which are made in a producer cell line after overexpression of a specific glycoprotein. And those Gesicles can be used to deliver proteins, for example Cas9, but also other macromolecular cargoes. And as the Gesicles also express VSVG on their cell line, their surface, sorry, they have a similar tropism as VSVG pseudotype lentivirus. So meaning this is a very broad mammalian tropism. So now what's our Gesicle production system? So this is really a do-it-yourself system to produce your own Gesicles, which will contain both the active Cas9 protein and your target-specific guide RNA. So how does the Gesicle production work? It's a bit similar to lentiviral packaging, and you will use a 293T based so-called producer cell line, which you will co-transfect with a plasmid, which will encode your target-specific guide RNA sequence, and then with a so-called Gesicle packaging mix. And in this packaging mix there's already our exact transfection reagent, which is contained, so it's a lyophilized mix. The mix also contains plasmids encoding the nanovesicle-inducing glycoprotein, plasmids encoding Cas9 endonuclease, and plasmid encoding membrane-bound red fluorescent cherry picker protein. So following transfection of the producer cells, the glycoprotein overexpression will induce the Gesicle budding from the producer cells. We also use a second technology in the system called idimerase, which is a technology to artificially induce protein-protein interactions. And we use this technology to tether the cytoplasmic Cas9 to the membrane-bound cherry picker protein. And this is just used to enrich the Cas9 and the guide RNA in the Gesicles, because naturally the Cas9 would not go to the membrane and would not be packed efficiently into the Gesicles. As you can see here, then the loaded Gesicles will just pinch off the producer cells and can be collected from the cell culture supernatant. And what's also nice is that you do not need to produce Gesicles each time you wish to do gene editing. You can also do a bigger production and then freeze them at -70 for later use. So once you will apply the Gesicles to the target cells, they will fuse with the target cells and will release the Cas9 protein together with the guide RNA into your cell. So here we are not speaking about DNA or RNA delivery. It's really a tool for Cas9 protein delivery to your target cells. So again, this is how the workflow looks like. What you will get in the kit are these 2 tubes with the so-called Gesicle packaging mixes, and what you will also get is a vector in which you can clone your target-specific guide RNA sequence. And you will then easily just add the guide RNA vector to the packaging mix 1, vortex the whole thing, and add this to the mix 2. And then you will transfect your producer cell line with the whole mix. Then remember, just to tether the Cas9 to the membrane-bound cherry picker protein, you will need to add the so-called AC heterodimerizer to your producer cells. This is to induce the artificial interaction of the Cas9 with the cherry picker protein and to enrich the Cas9 in the Gesicles. Then 2 to 3 days later, you can just collect the Gesicles from the cell culture supernatant, and then there is a centrifugation step to pellet and to concentrate the Gesicles, which are then ready to use. So once you will apply the Gesicles to your target cells, the red fluorescence will show you whether the delivery has been efficient. And you can also use the red fluorescence to enrich, for example, the positive cells using FACS. So, however, the red fluorescence will not really tell you whether you have delivered Cas9 and the guide RNA to your cells, because you could have delivered just empty Gesicles only containing cherry picker. So to check for the presence of Cas9 in your Gesicle production, there's one easy way. For example, you can just prepare a lysate and do a Western blot, for example, using our Cas9 polyclonal antibody. And then you can easily check for presence of Cas9 in your Gesicle preparation. For example, here, our R&D estimated that in 5 microliters of Gesicle preparation, there is about 200 nanogram of Cas9 protein. So as I told you in the beginning, the Gesicles also have VSVG on their surface, and that's why they have a very broad tropism. And so, of course, you can use Gesicles with easy-to-transfect cell types. For example, here, HEC or HeLa or MCF7 cells. And these data here show a side-by-side comparison with plasmid transfection. So in gray is the plasmid transfection, in blue the Gesicles. And we have measured here the efficiency of knockout of Z is green in stable cell lines. So this is measured on the protein level. And as you can see in this easier to transfect cell lines where plasmid transfection works very well, sometimes the Gesicle efficiency is a bit lower, sometimes it works a bit better, sometimes it's similar. So in all cases, however, you should get lower off-target effects. So even though sometimes maybe here Gesicle efficiency is a bit lower, you should get lower off-target effects compared to plasmid delivery because you will deliver an active protein which is degraded very rapidly. And I will speak about the off-target effects in a few seconds. Then, of course, the most interesting cell types are the more tricky cell types and the hard to transfect cell lines. And here really Gesicles can be a big advantage because you can see here on the right hand that the gene editing efficiency using plasmid delivery is almost inexisting or very low. And here, for example, in RPE, RAG, or Jurkat cells, we could really achieve correct gene editing efficiencies using Gesicles. Here from 20 to 40%. Our R&D has also successfully knocked out the CD81 gene in human iPS cells using Gesicle delivery. And here we have simply incubated the iPS cells according to the standard protocol, but with a bit longer incubation, so 6 or 24 hours, whereas the standard protocol is minimum of 4 hours incubation. And we have then measured the CD81 levels 6 days later, again with a FITC conjugated antibody. And as you can see, we could achieve over 40% of knockout of the protein level of CD81. The cell culture system we have used is our Cellatis FCS medium, which is a complete cell culture system for iPS cells. And what's very nice with the Cellatis FCS is that you can also do single cell passaging of your iPS cells and isolate single clones. So this is a very useful system to do gene editing experiments. Another interesting point is also that we have not observed any toxicity and no effect on the pluripotency when using Gesicles in iPS cells. So as you saw, the efficiency of the gene editing can vary depending on the cell line or the cell type you are using. Another important factor which has also an influence on the gene editing efficiency is the guide RNA scaffold you will use. And the scaffold which is included in our kit to clone in your guide RNA is an improved scaffold compared to the traditional scaffold that had been originally published. So the scaffold which is included in our kit has several mutations compared to the traditional scaffold. So the first one is UA flip, which will remove a premature termination sequence of transcription. And then this guide RNA scaffold here has also an extended hairpin. And this increases the stability of the guide RNA and will also improve the assembly of the guide RNA with the Cas9. And this is very important when you wish to pack both into your Gesicles to have a good assembly of Cas9 with guide RNA. And as you can see here at the end, also the gene editing efficiencies are much higher when using the optimized scaffold compared to the traditional scaffold. And that's why I would just like to really recommend to use the supplied vector, even though you might already have a guide RNA in a vector. But for efficient Gesicle packaging and efficient gene editing, it's important to use the improved scaffolds. Another factor which can influence the efficiency of the gene editing is also, of course, the amount of Gesicles you will add to your target cells. So in blue, again, you have results using the traditional scaffold, and in purple, results using the optimized scaffold. This is shown here for ACGFP knockout in a stable HT1080 cell line, again measured by flow cytometry 6 days after the delivery of the Gesicle, so on the protein level. So as you can see here in blue, increasing the amount of Gesicles when using the traditional scaffold does not really help, and you cannot really get over the 30% of knockout. However, when using the optimized scaffold, you can see here a nice increase also in the efficiency, and 30 microliters is what's currently recommended in the standard protocol. So for some cell lines, maybe 20 microliters can also be enough, but so this is the amount recommended in the standard protocol. So the last point I would like to discuss regarding the Gesicles are the off-target effects. And I would like to show you some side-by-side comparison with plasmid delivery for the EMX1 knockout. And so this, you can see here the sequence of the target site, EMX1, and there's also a potential off-target, which is shown here. So this experiment has been done in HEK cells, which are very easy to transfect. And as you can see here, plasma delivery here shown, so these lower bands indicate gene editing in a mismatch detection assay. So we could achieve about 16% of mutation frequency here in the population. But what you can also see here is that there are many gene editing events at this off-target site when using plasmid delivery. Now, in contrast, when using Gesicle delivery, you can see here gene editing at the EMX1 site. However, the off-target site is really clean and comparable to the negative control. This is also shown here on the sequence level. Our R&D has sequenced several clones. So again, on the left, the EMX1 site, on the right, the off-target site. Now here on top, you can see the plasmid transfection results. So you can see here the different gene editing events at the EMX1 site. And here, unfortunately, you can see that all the clones also carried mutations at the off-target site. Now, when you compare this to the Gesicle delivery, you can see that with Gesicles, you have gene editing events at the MX1 site, but that all clones here are really wild type for this off-target site. And this is really due to the fact that it's a method for protein delivery and that the Cas9 will not stay very long in the cell, but just for a few hours, and then it will be degraded. So to summarize for the Gesicle part, what are the major benefits of Gesicle delivery? So first of all, it's really suitable for a broad range of cell types, and these cell types also include difficult to transfect cells and non-dividing cells, for example, primary cells, and also, as I showed you, iPS cells. We have not observed any toxicity, also not in the iPS cells. And then really the major benefit is that Gesicles can be used to deliver active Cas9 protein. So first of all, you are not limited by promoters or codon usage, and then really you will avoid the persistent expression and the genomic integration of Cas9 into your target cells, and this really greatly decreases the off-target effects. Okay, so now the last step in the workflow is, of course, then to monitor downstream of the gene editing experiment whether your gene editing has been successful. And here I would like to present you 3 different tools which can be used. So these are the mutation detection kit, the genotype confirmation kit, and the indel identification kit. So let's start with the mutation detection kit. This is, I would say, it's a really quick and dirty method to quickly check your cell population for the mutation frequency. So as you remember, the efficiency of delivery of Cas9 is not 100% efficient, and also the efficiency of the gene editing is not 100% efficient. And so this means that at the end of your gene editing experiment, you will end up with a mixed population. And some cells will still be wild type, as shown here, and then the other cells will carry the different mutations created by the repair machinery of the cell. So now in the mutation detection kit workflow, it's also sometimes called the mismatch detection assay. You will, in a first step, amplify your target region using PCR. And what's nice with this kit is that it uses Terra polymerase for amplification, and Terra can also be used for direct PCR. So when using Terra, you do not need to purify your genomic DNA. You can just use a crude extract. And as you can see here, then after the PCR, of course, you will have a mixed population of molecules, with some molecules that will be wild type and some that will carry the different mutations. In the next step, you will simply denature and re-anneal the DNA, and all the molecules will be a bit scrambled, and this might create mismatches at some molecules. And the enzyme, which is included in the kit, the so-called guided resolvase, will specifically cleave this mismatched DNA. And this can then easily be analyzed on an agarose gel, and you can use densitometric analysis and a formula to check for the frequency of mutations in your cell population. I would like to show you some data compared to another enzyme, which is CEL1, which is also broadly used for these mismatch detection assays. And I would like to show you that our guided resolvase has better specificity. This is shown here. So this is our kit here on the left compared to the CEL1 enzyme on the right. And you can see that there's much less smearing when using the guided resolvase. And thanks to this higher specificity, we also have better sensitivity with our kit. So for example, when you have a look here in the lane 5, you can see a faint band here. So the legend of the figure is just transfected cells mixed with increasing concentration of non-transfected cells, meaning that here in lane 5, our kit is able to detect a mutation which is present in less than 20% of the genomic DNA pool. However, when you are looking here on the CEL1 assay, there's so much smearing that it's very difficult to say whether there was cutting or not. So better specificity and higher sensitivity of the guided resolvase. So now the second kit I would like to present you is the genotype confirmation kit. So the first kit, the mutation detection kit, is really a good tool for a quick screening of your cell population. This next kit here is a kit which can be used when you have already isolated clones derived from single cells. And this kit allows you to distinguish clones that are wild type from clones which carry monoallelic and clones that carry biallelic mutations. Again, the protocol starts with a PCR amplification of your target site, and again, this kit also uses Terra polymerase, so you do not need to purify your genomic DNA. And then it will be a simple in vitro cleavage with recombinant Cas9 and the guide RNA you used for the original gene editing experiments. And then you can imagine if, for example, unfortunately your clone is still wild type, for this in vitro cleavage assay, the original guide RNA can bind again and can guide the Cas9 to your target sequence, so you would see cleavage on the gel. In contrast, if your clone carries a monoallelic mutation, the original guide RNA could only cleave one copy and not the other allele. So at the end, you should get 3 fragments, 1 large and 2 small. And then if your clone carries a biallelic mutation, the original guide RNA should not be able to bind again, and you should see no cleavage at all. Just one remark, this kit cannot distinguish between heterozygous and homozygous mutations in your clones. So for all biallelic mutations, you would get 1 non-cut fragment, and then you would really need to do sequencing to determine whether a clone carries a heterozygous or a homozygous mutation. So this kit is more to pre-screen your clones quickly. For example, if you have a high number of clones and you don't have time to sequence all of them, you can quickly pre-screen and distinguish which clones are wild type, which are monoallelic, and which carry biallelic mutations. Just some results. So here for the C4 BTP editing, our R&D isolated 15 different clones derived from single cells, and they did the genotype confirmation kit. Just one remark again, it's always also important to include some wild-type DNA to check for proper cleavage of the Cas9 in vitro. If not, you could maybe end up with all non-cut fragments and you think it's all biallelic clones, but it's just the Cas9 that did not work. So it's always important to include some wild-type DNA to check for whether you get 100% cleavage here in the 2 small fragments. Okay, so now let's have a look at these 15 clones. As you can easily see here, 4 of the clones are wild type and can be sorted out. So no need to carry on those clones. Clone number 2 seems to carry a monoallelic mutation because there are 3 fragments. And the other clones here are very promising and seem to carry biallelic mutations. So now I've just put some of the sequencing results just below here. You can see that finally clone 2, which here was monoallelic, turned out to be a bit special because it had 3 copies of the gene. So 1 wild type and 2 different mutations. So this can sometimes happen when you're using a cell line which is not really diploid. And many of our cell lines which are broadly used in cell culture are not really diploid. So this could be the case. This could also happen if your clone was not originally isolated from 1 single cell, but was maybe a mix of 2 cells. So it's very important to have a clean clone at the beginning. Now the results for the other clones here, I've just put some examples. For example, clone 7, which here had 1 band, turned out to be heterozygous. And clone 9 and 11 turned out to be homozygous, and they had here, this one had a big deletion on both alleles, and this one here had an insertion followed by a deletion on both alleles. So to really distinguish whether a clone is homozygous or heterozygous, as I told you, you would need to do sequencing. And for this, we also have a kit, and this is the last kit I would like to present you. It's the so-called indel identification kit. So this kit is really a complete cloning system to recover the mutated sequences so that you can sequence them. This kit can be used either with a cell population to characterize the variety of indels which are present in your population, but also with single clones, and here then you can really determine the edits at the allelic level. So again, the first step of this kit is to amplify your target region using Terra polymerase, so again, crude extract for the PCR. And you will then clone the PCR product into the provided vector. And this kit uses our infusion technology. So it's a cloning technology which is not based on ligation, but based on homologous overlaps of the vector ends with the insert. And it's a very efficient cloning technology. And then you can transform the vector into bacteria and use the same Terra polymerase again just to do a colony screening, colony PCR, and then you can send this PCR product to sequencing, and you will see here, for example, you can see the different mutations here that occurred in a cell population. And you can also imagine if you start here with a clone derived from a single cell, and if this clone carries a homozygous mutation, then you should get here the same sequencing result in all of your bacterial clones. So just to summarize these 3 different downstream kits. The first kit we saw is the mutation detection kit, and this is really, as I said, a quick and dirty kit. It's a very quick evaluation of the mutation frequency in your cell population. So this one cannot be used with clones. It's really good for populations. It's very fast, 3 to 4 hours, and it allows you to see whether it's worth going on, for example, and select single clones. You can also roughly compare mutation frequencies from one experiment to another, but this kit will not allow you to see which mutations exactly occurred. It gives you more a rough percentage of efficiency. The second kit I presented is the genotype confirmation kit, and this one is only for single clones, and you can quickly distinguish wild type from clones carrying monoallelic and clones carrying biallelic mutations. It's also quite quick screening. I would say the whole protocol can be completed within 1 to 2 days, and this kit is really good if you don't have time to sequence all of your clones to sort out the ones that are not worth being sequenced. And then the last kit is the indel identification kit. This is really the kit with the highest workload because it takes 2 to 4 days or even longer, and it involves cloning and sequencing. But this kit will also give you the most detailed information because you can either evaluate the editing efficiency in a cell population and also characterize the variety of indels, or you can also determine the exact edits in single clones and at the allelic level. And just one last point, also don't forget about the Western blot or immunocytochemistry, really to check on the protein level whether you have obtained a functional knockout, because it's very important to sequence your clones and to check whether the desired mutation has occurred. But it's also really important to check on the protein level whether you have obtained the desired knockout. So I hope that I could show you that CRISPR-Cas is making gene editing much simpler and cheaper compared to other existing gene editing technologies. And I hope that I could also show you that our guided tools can help you throughout the whole workflow to produce your guide RNAs and to evaluate guide RNA efficacy in vitro prior to the delivery into cells. Then, of course, to efficiently deliver Cas9 and guide RNAs into mammalian cells, for example, using the Gesicles, or also in vivo, for example, using AAV. To quickly determine the mutation frequency in a cell population, to confirm the genotype of your clones, and finally also to determine the exact edits in single clones and on the allelic level by sequencing. I would also like to invite you to browse a bit on our website under www.clonetech.com/guided. And as you can see, there are really sections for each of the topics I discussed, and you can also find some technical notes online, and then of course the product pages. And that's good science. Yeah, thank you very much. I will now check whether there are any questions, and of course if you have questions later on, you can also just drop me an email to our tech support address. So let me just check your questions. Just 1 second. Okay, so we have one question. What is roughly the percentage of guide RNAs discarded after the in vitro testing? This is a bit difficult to say because sometimes all of them might function well. Here you could see in the example there was 1 out of 4. It's a bit comparable to siRNA. So in theory, of course, all guide RNAs should work because they are have homology to the target region. But in reality, sometimes depending on the GC content and the actual sequence of the guide RNA, this is not the case. But I cannot really tell you for each. It really depends on the sequence how many would be discarded or not. So here in our experiment, you could see it was 1 out of 4. So then we have a second question, whether there is a kit to test the efficiency of Gesicles in difficult to transect cells. We do not have a kit. What we have is we have ready-to-use Gesicles, which would be the easiest way to see just the efficiency of delivery. For example, we have ready-to-use Cre recombinase Gesicle that you could just put on your cells and then check whether they get red. Of course, this would not show you whether you would also get gene editing in those cells, but at least check the delivery. We don't have a real kit to test the efficiency, but if you don't want to produce your own Gesicles yourself for a first test, we have some ready-to-use Gesicles. We will also soon have ready-to-use Cas9 Gesicles, Cas9 only, that you could just put on your cells and check whether they get red. We have another question whether our kits can be used for prokaryotes. So our delivery tools, the Gesicles, our plasmids, and AAV are for mammalian cells only. But you could use our upstream and downstream kits. So the in vitro guide RNA testing could be used because it's a pure in vitro assay and you don't mind where the guide RNA sequence comes from. You could also use the mutation detection assay, for example, because the Terra enzyme can also work with bacterial samples, or also the indel identification kit if you want to sequence. So then we have another question, whether the system will only work with mammalian cells and not other cell types. So I believe you are talking about the Gesicles. So yeah, the answer is yes, it's mammalian cells. So if you're working here with bacteria or plants, then unfortunately Gesicles would not be suitable. Then we have another question, whether it's possible to deliver a repair template for homologous recombination at the same time with the Gesicles to do the knock-in. So yeah, this is a very interesting question. And for the moment, unfortunately, we don't have a method to pack a repair template into the Gesicles. Because remember, the Cas9 and the guide RNA will naturally form a complex. And then you will tether the Cas9 to the cherry picker to enrich this. But if you would just put a repair template as plasmid or as a single-stranded DNA oligo, they would not be efficiently packed into the Gesicles. So for the moment, we don't have a protocol to get this into the Gesicle, and you would need to deliver the repair template separately. So meaning you would need to use electroporation or transfection. And then the question is more, what's the best time point to deliver the repair template? So this would be, you would need to determine this empirically to see whether it's better before or after the Gesicle delivery. Then another question about supplies in South Africa. Yes, I will contact you by email and give you the address of our distributor. Then a question is whether anybody tried, I believe, the Gesicles and primary neurons. So in-house, we have not tried this, but I would say, so first of all, primary cells can be used. You do not need proliferating cells to deliver Gesicles because it's VSV-G mediated fusion with the target cells. Now, yeah, I would really say as long as your cells maybe express the LDL receptor, which has been described as one entry point for VSV-G, this should be fine. And I would say, yes, it should work in neurons. The cells which are the most tricky to deliver with VSV-G pseudotype lentivirus or Gesicles are more hematopoietic stem cells, B cells, or yeah, non-stimulated B cells and T cells. So for neurons, I would say it would be worth giving it a try. Another question again is whether it has been tested in primary keratinocytes, sorry, and it's a bit the same answer. We have not tested it in-house, but I think it should work. Let me just check whether there are more. One question for the plant cells. So yeah, whether we are developing kits for plant cells. So unfortunately for the delivery part, no, we don't have anything for plants or bacteria. It's all for mammalian. What you could use again are the upstream screening kits and the downstream mutation detection or genotyping kits. Because the Terra enzyme works with plant samples. But for the delivery part, for the moment, we don't have anything. One last question, whether the generated guide RNAs could be used to transfect other types of cells, for example, plant cells. So here, I believe you're speaking about the guide RNAs used produced by in vitro transcription. So those can also be used for transfection. I think we have tested this in mammalian cells, but you could also, if you can get those into plant cells with a method, this could also be active in plants. Okay, so that's all for the questions. I would like to thank you again for attending the webinar, and we will then send you the presentation as PDF copy by email. Thank you very much and have a good day.